for breaking news Davao.
Good morning, Philippines. Good morning, Davao del Norte State.
It is a sunny day, April 25, 2022.
I am Thea Tabalanza.
The Beata Palace.
The breaking news. For the headlines,
Glucose can be synthesized from non-carbohydrate precursors.
Gluconeogenesis and glycolysis are reciprocally regulated.
Metabolism and contacts. Precursors formed by muscle are used by other organs.
Gluconeogenesis is the synthesis of glucose from non-carbohydrate sources, such as lactate, amino acids, and glycerol. For the international report, we have Alana Tampo.
Gluconeogenesis. Gluconeogenesis is a synthesis of glucose from non-carbohydrate precursors. Why gluconeogenesis is important? It is important for fasting and starvation because it maintains blood glucose level and concentration and glucose required by brain. Major site of gluconeogenesis is the liver and will taking place within a small amount in the kidney. And gluconeogenesis and glycolysis are opposite pathway, but not complete reversal of glycolysis. Why? Because glycolytic steps involve transferring a phosphate group from ATP, and gluconeogenesis can't generate ATP. There are three gluconeogenic precursors, which are the lactate, the amino acid, and the glycerol. In glycolysis, glucose is conversion to glucose 6-phosphate by the enzyme hexokinase. Fructose 6-phosphate converted to fructose 1,6-biophosphate by the enzyme phosphofructokinase 1, and the last irreversible step, which is the conversion of phosphoenol pyruvate or PEP by the enzyme pyruvate kinase. So, when we are trying to reverse this pathway, which is the glycolysis pathway, kani siya gikan sa glucose nga mag-end sa pyruvate, which means it is a glycolysis pathway. When we are trying to reverse this pathway back up to glucose gikan sa pyruvate, it means nga mao na pud ni siya nga to ang gluconeogenesis pathway. So the beginning of the pathway simply is the end of glycolysis pathway, which is a pyruvate. Now, in order to go around the enzyme pyruvate kinase, pyruvate has go through the enzyme pyruvate carboxylase to form oxaloacetate. So, asa man ni nalocate ni si pyruvate carboxylase? So, si pyruvate carboxylase nalocate ni siya into mitochondria. Now, natay oxaloacetate from the pyruvate carboxylase, no? So, kanina po si oxaloacetate will also have to be converted using a different enzyme to phosphoenol pyruvate by the enzyme PEP carboxykinase or phosphoenol pyruvate carboxykinase. So, we see that these two enzyme, kani siya nga mga enzyme, required an equivalent of 2 ATP. So, as we go backward in a pathway from phosphoenol pyruvate, kaya naman tayo phosphoenol pyruvate, mubalik ta back to 2,2-phosphoglycerate to 3-phosphoglycerate. So in the reversal conversion of 3-phosphoglycerate back into 1,3-biophosphoglycerate, we have to use the enzyme phosphoglycerate kinase, which is actually cause another ATP. Nagkinahanglan na po na siya equivalent of another ATP, kani si phosphoglycerate kinase, which is actually cause another ATP na, diba? So, once nga naa natay 1,3-biophosphoglycerate, this can be converted back to glyceraldehyde 3-phosphate. Then, ang kani si glyceraldehyde 3-phosphate, which it can be converted back to fructose 1,6-biophosphate. Then, kani si biophosphate na pod will be converted to fructose 6-phosphate by the enzyme fructose 6-biophosphatase. Then, kani si fructose 1,6,5 fructose 1,6-phosphate will be converted back to glucose 6-phosphate, and glucose 6-phosphate will be converted back again to the glucose by the enzyme of glucose 6-phosphatase. Another information, this gluconeogenesis pathway has equivalent of 6 ATP needed for this pathway. It's because phosphatase are the enzyme required for this reversal step in gluconeogenesis, which is total amount of ATP in the step in gluconeogenesis pathway, actually twice the amount of ATP, because what we do is 2 ATP used in pyruvate carboxylase and PEP carboxykinase, and another one for the phosphoglycerate. So the first back into gluconeogenesis pathway, the first precursor for gluconeogenic pathway is the lactate. So giunsa man sa atong mga precursor nga mag-get into gluconeogenesis pathway. So the first is the lactate. So through co-recycle, kani si lactate will be brought into the liver, into hepatolysis, undergo the enzyme lactate dehydrogenase to form pyruvate. And this enzyme, kani si lactate dehydrogenase, will actually require a NAD+, and actually reduce the NAD+ to NADH, and that also shunts lactate to gluconeogenesis pathway. The second one is the amino acid. There are many different gluconeogenic amino acid. Some are these kay maano siya, so si alanine, si cytosine, si glycine, si serine, si threonine, o si tryptophan. So kani nga mga amino acid will be directly processed to gluconeogenesis pathway. And another amino acid nga can be processed to oxaloacetate is the asparagine. Asparagine can be processed to asparaginase into aspartate. Then aspartate can be processed oxaloacetate by the enzyme nga kani si aspartate aminotransferase. Then another na pud nga mga amino acid kay mao ni si arginine, si glutamate, si glutamine, si histidine, si phenylalanine, si proline, si isoline, methionine, threonine, tyrosine, si valine, which is kanina pud nga mga amino acid can be shunted into TCA cycle, kani si TCA cycle, then will be directed to oxaloacetate, which can be used for gluconeogenesis pathway. So through oxaloacetate makasulod na nga tong amino acid gikan ka oxaloacetate paingon sa gluconeogenesis pathway. So the third precursor will be the glycerol. So glycerol will be developed in the parasite in the liver, will be phosphorylated by the enzyme glycerol kinase. This enzyme required ATP. So si glycerol kinase will form a glycerol phosphate, which is the glycerol phosphate na pod will be dehydrogenated by the enzyme glycerol phosphate dehydrogenase to form dehydroxyacetone phosphate. Then kani si dehydroacetone phosphate can be shunted into gluconeogenesis pathway. Then kani si glycerol phosphate before na kumalimtan naga-require siya NAD+ and reduce NADH. There are 3 essential irreversible steps in glycolysis pathway, which are the first is glucose plus ATP by the enzyme hexokinase form a glucose 6-phosphate plus ADP. And the delta G for this is negative 33 kJ mole raised to the negative 1 and negative 8.0 kcal mole raised to the negative 1. The second is a fructose 6-phosphate plus ATP by the enzyme phosphofructokinase turns to fructose 1,6-biophosphate plus ADP. So the delta G for this is negative 22 kg mole raised to the negative 1 and negative 5.3 kcal mole raised to the negative 1. So the last irreversible step, which is the phosphoenol pyruvate plus ADP by the enzyme pyruvate kinase turns to pyruvate plus ATP, and the delta G for this is negative 17 kg mole raised to the negative 1 and negative 4.0 kcal mole raised to the negative 1. In gluconeogenesis, the following new step bypass this virtually irreversible reaction of glycolysis. First is the phosphoenol pyruvate is formed from pyruvate by the way of oxaloacetate. Second one is the fructose 6-phosphate is formed fructose 1,6-biophosphate by the hydrolysis of the phosphate. Third is the glucose is formed by the hydrolysis of glucose 6-phosphate.
So as I've said, sa giingon nako ganina nga ang pyruvate carboxylase is na located sa mitochondria. So that means that pyruvate has to enter the mitochondria will actually be processed by an enzyme. So gyud siya pagbuhat. The first thing that has been done is that pyruvate has been translocated in the mitochondria, and sa pamamagitan ani na kaning transporter man ni siya si mitochondria pyruvate carrier. Mao ni siya ang magdala ng pyruvate paingon diri sa mitochondria. Once nga ang pyruvate kay naa na sa mitochondria, ang kaning pyruvate na pod gina-process na pod siya sa pyruvate carboxylase into oxaloacetate. And now this enzyme requires or activated by the acetyl CoA, and it requires ATP. Also, nag-require po ang enzyme og biotin or vitamin B7 para mo functioning pyruvate carboxylase to form oxaloacetate. So naanatay oxaloacetate. Nya kun sa oxaloacetate can be formed to malate by the enzyme malate dehydrogenase. So makita nato nga naay duha ka arrow, which is reversible step ni siya nga ang malate pod pwede po ni siya ma-form into oxaloacetate with the reversible enzyme malate dehydrogenase. So and the cofactor for this enzyme is NADH. Since in mitochondria it has a higher level it has a higher level of NADH. So ang kanipong NADH will actually be processed and utilized to form NADA+. Since nga nanatay malate can penetrate or exit in the mitochondria membrane because of malate can penetrate or exit the mitochondria. So imagine na nato nga kani nga part kani diri kay mao ni siya nga tong mitochondria nya diri sa baba kay mao ni siya tong cytosol. So kani si malate mo makagawas ni siya sa mitochondria membrane sa pamamagitan ni malate aspartate shuttle. So once nga naa na diri si malate sa ato ang cytosol, ang malate na pod can actually convert into oxaloacetate. By the enzyme maura gyapon si malate dehydrogenase. So mao na akong giingon ganina nga pwede nato siya i-reverse ang kaninga process. Pero diri sa cytosol ang NADA+ ang mas daghan kaysa sa NADH. Then oxaloacetate can be processed by the enzyme phosphoenol pyruvate kinase or PEP kinase. And kaninga enzyme naga-release ni siya og CO2 molecule, and this enzyme requires GDP to form for its function rather. So we ended up having phosphoenol pyruvate by PEP carboxylase by the enzyme PEP carboxykinase. How fructose 1,6-biophosphate processed into fructose 6-phosphate. I said earlier that
it is processed by the enzyme fructose biophosphatase. In doing so, fructose 1,6-biophosphate give converted into fructose 6-phosphate by the enzyme fructose 1,6-biophosphatase. And it actually releases PI, and these steps, transitional steps, does not cause any ATP and energy. Now, this is the important enzyme because it is actually regulated, and it's regulated by the glucagon. Actually, glucagon activates fructose biphosphate, and it actually activated by the citrate. And this enzyme inhibited by AMP and inhibited again by fructose 2,6-biophosphate. So, asa man gikan si fructose biophosphate? Gikan ang fructose 2,6-biophosphate gikan siya sa conversion of fructose 6-phosphate by the enzyme phosphofructokinase 2 or the PFK2 rather. And remember, in glycolysis pathway, it is actually PFK1 that produces fructose 1,6-biophosphatase, and phosphofructokinase 2 regulated by glucagon. Kani siya na-regulate gyapon siya sa glucagon, which is actually inhibit by the PFK2. Actually, kani si glucagon does two things, and it is important step in the gluconeogenesis. Dili man nimo activate ni si fructose 1,6-biophosphate kung wala ang glucagon. Once we have a fructose 6-phosphate na it actually converted into glucose 6-phosphate by phosphoglucose isomerase. Actually, kani diri nga part kay diri ana ang interesting nga part, which is the next step happens in the endoplasmic reticulum kani siya. Diri endoplasmic kunuhay ni glucose 6-phosphate became unphosphorylated inside the endoplasmic reticulum. So giunsa siya pagbuhat nga na phosphorylated na siya. First thing is that glucose 6-phosphate, kung si glucose 6-phosphate i-transfer ni siya, will get transferred into endoplasmic reticulum through transporter 1 or what we call T1. So kani si T1 ang magdala sa glucose 6-phosphate dito sa reticulum. So glucose 6-phosphate nag-enter na siya sa endoplasmic reticulum, and glucose 6-phosphate is actually located in endoplasmic reticulum. Now, this is important to know. Glucose 6-phosphatase, kasi glucose 6-phosphatase only located in the liver, and it's small amount in the kidney. That's why the liver and the kidney are the only organ plays a role in gluconeogenesis. Back to this, glucose 6-phosphate will actually converted into organic phosphate, and glucose is within the endoplasmic reticulum na, and they are both actually transport in a different transporter out in the endoplasmic reticulum. So the T2 or the transporter 2 for inorganic phosphate, and T3 for glucose, they both transfer out in endoplasmic reticulum to cytosol.
That's all. Thank you. Back to you, Thea.
Thank you, Alani. Meanwhile, gluconeogenesis and glycolysis are reciprocally regulated, so that one pathway is relatively inactive while the other is highly active. For more updates, we have Nielsen Glucopolis for the National Report.
Gluconeogenesis and glycolysis are reciprocally regulated. The basic premise of the reciprocal regulation is that when glucose is abundant, glycolysis will predominate. When glucose is scarce, gluconeogenesis will take over. However, the activities of the distinctive enzymes of each pathway are controlled, so that both pathways are not highly active at the same time. The rate of glycolysis is also determined by the concentration of glucose. and the rate of gluconeogenesis is controlled by the concentration of lactate and other precursors of glucose. Energy charge determines whether glycolysis or gluconeogenesis will be more active. The figure shows during low starvation. In this case, the concentration of AMP is high. Under this condition, AMP stimulates phosphofructokinase but inhibits fructose 1,6-biphosphatase. Thus, glycolysis is turned on and gluconeogenesis is inhibited. Conversely, high levels of ATP and citrate indicate that the energy charge is high and that biosynthetic intermediates are abundant. Under this condition, glycolysis is nearly switched off and gluconeogenesis is promoted. High levels of citrate indicate an energy-rich situation in the presence of precursors for biosynthesis, most notably fatty acid synthesis. The second figure it shows during starvation. Glycolysis and gluconeogenesis are reciprocally regulated at the interconversion of phosphoenol pyruvate and pyruvate in the liver. Conversely, pyruvate carboxylase, which catalyzes the first step of gluconeogenesis from pyruvate, is inhibited by ADP. Likewise, ADP inhibits phosphoenol pyruvate carboxykinase. Pyruvate carboxylase is activated by acetyl, which, like citrate, indicates that the citric acid cycle is producing energy and biosynthetic intermediates. Hence, when the cell is rich in biosynthetic precursors and ATP, anabolic pathways are favored. In the liver, gluconeogenesis can produce glucose to replenish glycogen stores, and fatty acid synthesis can take place. Balance between glycolysis and gluconeogenesis in the liver is sensitive to blood glucose concentration. In the liver, rates of glycolysis and gluconeogenesis are adjusted to maintain blood glucose level. As you can see in the figure, the purple is the kinase domain fused to the red, which is phosphatase domain. The bar represents the amino acid sequence of the enzyme. This bifunctional enzyme contains an N-terminal regulatory domain, followed by kinase domain and phosphatase domain. Glucagon stimulates pKa when blood glucose is scarce. FB phase 2 is activated, glycolysis is inhibited, and gluconeogenesis is stimulated. Gluconeogenesis predominates. Glucose formed by the liver under this condition is essential for the viability of the brain. Glucagon stimulation of protein kinase A is also inactivates pyruvase kinase in the liver. Conversely, when blood glucose is abundant, as if it after meal, glucagon concentration in the blood falls and insulin levels in the blood rise. A low blood glucose level is signaled by glucagon leads to phosphorylation of the bifunctional enzyme and hence to a lower level of fructose 2,6-biphosphate, slowing glycolysis. High levels of fructose 6-phosphate accelerate the formation of fructose 2,6-biphosphate by facilitating the dephosphorylation of the bifunctional enzyme. Clinical insight. Untreated type 2 diabetes can progress to insulin-dependent diabetes. The treatment of type 2 diabetes include weight loss, healthy diet, exercise, and drug treatment to enhance sensitivity to insulin. The figure shows a substrate cycle. This cycle, the ATP-driven cycle, operates at two different rates. A small change in the rates of the two opposing reactions result in a large change in the net flux of product B. A pair of reactions, such as the phosphorylation of fructose 6-phosphate to fructose 1,6-biphosphate in the glycolytic pathway and its hydrolysis back to the fructose 6-phosphate in the gluconeogenetic pathway is called substrate cycle. Despite such extraordinary circumstances, substrate cycles now seem likely to be biologically important. One possibility is that substrate cycles amplify metabolic signals. This amplification is made possible by the rapid hydrolysis of ATP. The flux of each step of the glycolytic pathway has been suggested to increase as much as 1,000-fold at the initiation of intense exercise, when a lot of ATP is needed, because the allosteric activation of enzymes alone seems unlikely to explain this increased flux. The existence of substrate cycle may partly account for the rapid rise of the rate of glycolysis. That's all, and back to you, Thea.
Thank you, Mieza. Now, let's move on for the local report. Metabolism and context. Precursors formed by muscle are used by the other organs. Based on the previous discussion of Ms. Alani, she tackled about the gluconeogenesis precursor, which is the lactate, glycerol, and amino acids. In gluconeogenesis, the primary occurs in the liver and secondary occurs in the kidney. Lactate, produced by active skeletal muscle and red blood cells, is a source of energy for other organs. Red blood cells lack of mitochondria and can never oxidize glucose completely. In contracting skeletal muscle during the vigorous exercise, the rate at which glycolysis produces pyruvate exceeds the rate at which the citric acid cycle oxidizes it. In the cells, lactate dehydrogenase reduces excess pyruvate to lactate to restore redox balance. In contracting skeletal muscle, the formation and release of lactate lets the muscle generate ATP. In the absence of oxygen and shifts the burden of metabolizing lactate from muscle to the other organs. In the previous discussion about metabolism, we all know that lactate is a dead end in metabolism. It must be converted back into pyruvate before it can be metabolized. Lactate can diffuse out of these cells through carriers into the blood. Lactate in the bloodstream has two fates, which is plasma membrane of some cells contain carriers that make the cells highly permeable to lactate. Number 2, excess lactate enters the liver and converted first into pyruvate and then into glucose by the gluconeogenic pathway. In one fate, the plasma membranes of some cells, particularly cells in the cardiac muscle, contains carriers that make the cells highly permeable to lactate. The molecule diffuses from the blood into such permeable cells inside these well-oxygenated cells. Lactate can be reverted back to pyruvate and metabolized through the citric acid cycle and oxidative phosphorylation to generate ATP. Also, the use of lactate in the place of glucose by these cells makes more circulating glucose available to the active muscle cells. In the other fate, excess lactate enters the liver and it's converted to pyruvate and then into glucose by the gluconeogenic pathway. Thus, the liver restores the level of glucose necessary for active muscle cells, which derive ATP from the glycolytic conversion of glucose into the lactate. So this reaction constitutes the Cori cycle. Alanine, like lactate, is a major precursor of glucose in the liver. The alanine is generated in muscle when the carbon skeleton of some amino acid are used as fuels. The nitrogen atoms from this amino acid are transferred to pyruvate to form alanine. The reverse reaction takes place on the liver. This process also helps to maintain nitrogen's balance. If you observe this figure between the gluconeogenesis and glycolysis, if you observe they have a P symbol. The P symbol represents the nucleosides triphosphates, and this pathway help meet the energy needs of different cell types. In this discussion, always remember that lactate is generated by glycolysis and contracting muscle is released into the bloodstream. This lactate is removed from the blood by the liver and is converted into glucose by gluconeogenesis. This metabolic cooperation between muscle and liver is called the Cori cycle. Lastly, Alanine is used to transport nitrogen as well as carbon skeleton from the muscle to the liver. That's all, and thank you. I am Thea Tabalanza.
The Beata Palace.
The breaking news.